Thank you. Well, good afternoon, everybody, and it's a pleasure to join you, and particularly to listen to Frank Dana. Frank is professor of computer science, but his research has really crossed disciplinary boundaries, and that's what he's going to talk about today. You know, we all hear about proteins, which are all over the place, and they really regulate a lot of life, but some non-natural ones can be designed for a specific purpose, and that's the sort of thing that Frank is interested in. So Frank is the founding director of the Institute for Data Science here at Carleton, which has been going since 2004. And 2014, and we have a number of large events. In particular, is the Data Science Day, which is available for registration on our website. So please join us. We've got a great program. So with that, any further ado, Frank, I'll pass this to you because this is all about you this afternoon.
Thank you so much, John. So thank you very much to everybody for joining our little afternoon session here. And thanks, John, for the invitation. So so my name is Frank Dana, and I'm a professor at Carleton University in the School of Computer Science. And like as John just pointed out, I have multiple hats. So I'm also the founding director of our Institute for Data Science. I'll talk about that in a second, which is an interdisciplinary institute for AI and data science. I'm also cross-appointed to the IBM Center for Advanced Studies in Toronto. So my research is like three-pronged. Like my background in computer science is about parallel computing. So that's the kind of classes also I teach, and that's my background training is in programming those large multi-core clusters, or these days they're called clouds, like large-scale cloud computing. And really, I'm into algorithms for those things, how to program big clusters and clouds, like something with 40,000 processors, not something you put on your desk. I'll show you some pictures in a moment. And now to use those big machines to do things that are useful. And so the two areas I'm concentrating on are things in data analytics, and that's mainly linked to my work at IBM, and computational biochemistry, protein-protein interactions, and that's what I want to talk about today. And that's a research group here at Carleton. So before I get into that, just again, just a little bit of a background about the Institute for Data Science. We started in, John, I think it's 2013, if I'm correct, or 2014. It took us a while to get everything started. And it started actually with a seed grant from IBM. And we've hired a whole bunch of professors in the meantime, and running a rather large graduate program. My slide says 120 master students, but it's actually by now even more. I think we've hit 150 by now. So in terms for graduate student, that's a very large number. And we're starting a PhD in data science this fall. And it's truly interdisciplinary. You see on the right side all the departments that participate in our program. So it's not just computer science, computer engineering, but computer science is everywhere these days, right? That's nothing new. So it's graduate students in biology and business, communication, cognitive science, economics, geography, health science, history, psychology, public affairs, public management, who do research in data science. So my definition of data science is really is very simple when I talk to students. It's computer scientists working with people in other fields to help them do useful stuff. That's my kind of simplified definition of data science and AI. So our project here that I want to talk about today is using these big machines, as that you can see on the left. Like this is a big IBM supercomputer, the kind of machine I'll talk about later. It has about 40,000 processor cores, sits in Toronto, and is a shared resource for researchers in Ontario. And we are using this machine to design algorithms to figure out what proteins do. So in particular, protein-protein interactions. And so I want to take a few minutes to talk about what that is. But before I do that, I also want to make sure everybody knows that this is a team effort that I'm talking about. So it's not just myself. It's a whole team of people that's been working on what I'm talking about since 2003. So time flies. It's a very long journey. I mean, it's, you know, science is very work intensive. And so there's a whole bunch of professors from different departments, like particularly in biochemistry, my colleague Ashkan Golshani, and his previous supervisor, Jim Greenblatt in Toronto at the Bunting and Best Institute, and Jim Green in biomedical engineering. And on the right side, you see a whole bunch of graduate students who did their masters and PhDs on this topic, both in computer science and biochemistry. So this is a large group effort over a long period of time. And so it's a little bit challenging also for me to present all of this in a few minutes. So the background is, I mean, most of you probably know about genomes, right? So each of our cells has a DNA, right? And the DNA sits in the nucleus and it just sits there. It's essentially like a piece of text. And what happens is these, the DNA, you know, are codes for amino acids of proteins, right? So always it's essentially a character sequence, right? We have 4 different characters, GCTA, and always triples of these characters actually code for 1 amino acid. And so the RNA is kind of what's called the messenger RNA, is the way of taking this information, you know, out of the nucleus and bringing it into making proteins out of this. So the first step is what's called transcription going on in our cells, where pieces of DNA, like one gene which codes for 1 protein, is translated into this RNA sequence. And then the messenger RNA travels through the cell, and then eventually a protein is created out of that sequence. Right? So this is how proteins look like. On the right, you have a picture of kind of how they kind of really look like. On the left, you have the stick figure. And in the middle, you have some intermediate representations because we do have some secondary structures. But really what it is, I mean, a protein, it's a linear sequence of these amino acids. And, you know, they get, you know, really chemically created. So we have this long molecule, which is a sequence of amino acids. And then like a string, it kind of folds into a ball. And that's what you see on the right side. And what is important about proteins, what's really amazing, is it folds into a very stable structure. So, I mean, at least the proteins we have. And so they have a very distinguished shape. Each protein has its own shape. And that shape is extremely important because now the shape of these proteins determines how different proteins work together. And it's kind of like a lock and key operation going on in our cells. You see on the left side, you have the blue protein and the green protein. And they have this yellow area there on the blue protein, which matches the yellow outside area of the green protein. And it's like a lock and key mechanism whereby now these two areas fit, right? And so those two proteins can dock using the yellow area. And it's really like a key fitting into a lock. It's a very, very precise mechanism. And so proteins are very particular with whom they dock. And on the right side of this picture, you see more like the biochemist's view of this with more details. So now these protein interactions are really what runs our body. Each cell, like humans have about 20,000 different genes, so that code for 20,000 different proteins. And now which protein interacts with whom really determines mechanisms, processes going on in our cell. The picture you see here is the picture for yeast, right? So yeast is a bit simpler. It has 6,000 proteins. And, so you see here how the genes, how the proteins made by these genes are clustered into groups of proteins interacting with each other. And these groups of proteins, they're called pathways. They are responsible for certain functionalities in our cells, right? So this is really the mechanism of life going on in each and every one of us. And it's, of course, very important to figure out this mechanism, right? So to just draw this picture is an awful lot of work. So chemists, they test which protein interacts with which one through a process called tap tagging, where they literally check, like, for example, in this case, we are checking whether this yeast, this is for yeast, the yeast protein YGL227W, whether it interacts with this other protein, YMR135C. And the way you do this is by effectively grinding up the yeast cell, making sure we have a free-floating bunch of proteins, and then pulling out the YGL and checking whether it also pulls out the YMR protein. And that's what you see here in the gel. And then you try it the other way around. You pull out the YMR and check whether it also pulls out the YGL. And if it happens if they happen to both show up in your experiment, then you say, oh, well, these two guys interact, they stick to each other. 1 second, please, I have to close my door. Okay, so now this experiment takes a day or two, and as I just said, we humans have 20,000 different proteins, so that's 200 million combinations times 2 days. So you can see that this is a very lengthy, if not impossible, task. And here you see also for other species, like for yeast or for like a little worm. Interestingly, the little worm has more proteins than humans, right? So for humans, by now, we know about 40-something thousand interactions. And, biochemists have been working hard on that. But, like my friend Ashkan, he did his PhD, Ashkan Golshani, right, my collaborator in biochemistry, he did this PhD on this kind of stuff. And we literally met in the pub, in the graduate pub, and he was kind of tired of running these experiments. And we were thinking about, can we do something computational to speed this up, right? So in terms of computer science, we by now have a list of these 40,000 plus known interactions. So we know which proteins interact. So now I give you 2 new proteins and check, do they interact? That's the question, right? Proteins I haven't tested yet. And it's actually kind of a classical machine learning thing, right? It's like, on the web, you show an AI algorithm 20,000 cats, and then you show it a new picture and the algorithm tells you, oh yeah, there's a cat, right? So it's a similar kind of problem. And so here is our algorithm that we came up with. So I'm not going to bore you with the details of, the coding and the implementation of this. I'll rather show you what it does. So here is what our AI method does to detect, to predict whether two proteins interact without running an experiment. So effectively, what you see in this picture here is on one axis, the YGR protein has 800 amino acids. So that's the length of the protein. On the other axis, the YBR protein has 500 amino acids. Okay. So now on this plane here, you have represented all the combinations of locations in protein YGR and YBR. And we're looking at little windows at these combinations of locations. So let's say we look at a window which is at location 300 in YGR and at location 200 in YBR. Then now for the Z-axis, we measure a number. So what is this number? So what we calculate effectively, and this is a little bit simplified, but the basic idea is for each such combination of locations in YGR and YBR, we look into all the known data, right? Oops, sorry. These 40,000 known interactions. And we calculate how many pairs of interacting proteins that we know do interact are out there. So how many pairs of proteins AB are out there which contain in A little subsequence that is very similar to what we find at location 300 in YGR, and in location B, it contains a little subsequence that's similar what we find at location at location 200 in YBR. So this is the value for what we call hits for the Z-axis. We call these co-occurrences, right? So what you see here in this picture is this peak. So what this peak means is that at this combination, location 780 in protein YGR and 150 in YBR, if you look at those two little subsequences, these little windows of amino acids at these two locations, there is 100 pairs of proteins that are known to interact, where one protein contains a window that's very similar to what we have at this location here in YGR, and the other contains a subsequence that's very similar to what we have here in YBR. So we effectively have a very high number of indications that, oh, maybe this is an interaction site. And so this would be a positive result if we find such a peak. And a negative result would look like this. We don't find such a peak. And so if we find such a peak, then we predict that, yep, these two proteins most likely interact. And the height of the peak kind of represents the probability, which we think they do interact. So it's all probabilistic. But at the end of the day, now there's a lot of details like what height of the peak do you need to say, yep, they interact and stuff like that. So here, I mean, at the end of the day, we can predict whatever we want. The question is, does nature agree with us, right? Because if nature doesn't agree with us, whatever we do is totally irrelevant. So here we have, in this example, two proteins, again, yeast proteins. It's easier to experiment with yeast proteins than with humans, right? I mean, so we have these two yeast proteins here that have never been tested in the lab. And so we ran our method and we find actually two peaks even, and they're quite high. And so we say, oh, wow, these guys interact. So then we sent it off to Toronto, to the Bunting Best Institute, and they did the experiments. And wow, they actually do interact. So that's cool, right? And so we've done a lot of these experiments. We've done a lot of experimental validation of this method. And I'm not going to go into the details of this, but those who are expert in statistics, it's these kind of what are called ROC curves that represent the quality of the prediction. And it turns out that our prediction method, and what I showed you was a very simplified thing, right? Version of it, but there's lots of details to still work out. But at the end of the day, our prediction method ended up being very precise. And of course, we're not the only ones doing this, right? I mean, this is an extremely important problem. So there's even competitions out there. So one of the scientific journals, BMC Bioinformatics, they ran a competition of people who do these protein interaction predictions. And, people sent in their code. They run it against protein pairs which they know interact and which they know don't interact. And they just look at your hit rate, right? How often are you correct? And interestingly, what turned out is that our method was by far the winner, and not even close. Like the precision of the runner-up was less than a third of our precision, right? So it wasn't even close. So since then, we've been extremely busy. So we've had lots of projects in various medical science institutes who want to know protein interactions for all kinds of things, like diseases of all kinds. And of course, the other thing we want to do is create this graph, right? My friend Ashkan said, okay, Frank, so now that you know how to predict protein interactions for one protein, please, you have a computer, run it on all combinations. And I had to explain to him that, well, that may take a while. And I had a PhD student who actually, he did, we call it the PhD with the largest carbon footprint, because he ran a processor with 1,000 cores for 3 months, 24/7, to run this algorithm on all 200 million combinations of human proteins. And he found with a very high, high precision, very high probability interactions, he found about 130,000. So that more than quadrupled the number of known protein interactions. And so, for example, here's what people do with that kind of knowledge. So we have DNA break repair proteins. We have a mechanism in our cell to protect us from cancer. For example, we all are constantly exposed to radiation from outer space, right? Not just artificial radiation, but natural radiation. And because of that, all animals on Earth have these anti-radiation protection mechanism. And it's called DNA break repair, right? So the radiation, damages our DNA, and there's a bunch of proteins who constantly go around and fix our DNA so we don't get cancer. It doesn't always work, as we know. People do get cancer, but without them, we wouldn't be living very long. And people, of course, want to really understand how this works. So in this picture on the left, you see the blue proteins which were known to be involved in this DNA repair. And when we calculated our expanded graph of interactions, right, these 140,000 or 130,000 additional interactions, those are the red edges here. These are new edges, new interactions that we discovered. When you look at the picture of the blue guys, you see those 3 yellow guys there, which were previously unknown. Nobody knew that they are actually also involved in cancer prevention. And actually turns out people did experiments, not on humans, but on the equivalent proteins in yeast. And when you put yeast under radiation and they are missing even one of those yellow proteins, they die right away. So so just one example of many for these protein interaction prediction to just give us more knowledge. And again, like this has been such a long-standing project. I could keep talking about this for a long time, many other projects. But what I want to actually switch to now is something different, is using our algorithm that predicts protein interactions, now not just to study proteins, but to design new synthetic proteins, artificial proteins that do certain things, that have specific protein-protein interactions, which actually change things in our cells and which can be used as medication. So this is a well-studied area in the, when you look at drug design, right? So if you look at, our big drug companies, they're in this business. So because remember I was saying earlier, our cell has these clusters of proteins, which are called pathways, which interact with each other within the cluster in a certain way, and that creates a certain functionality. And many diseases are caused by one or more of these pathways being wrong, right? Not working properly. So now you want to kind of throw in something that interacts with them and makes things right. And in the vast majority of cases, it's actually these pathways are overdoing things. So the vast majority of drugs are actually down-regulating pathways. And so the question for synthetic proteins as medication is like this. So we have a pathway, and within the pathway, we have a protein or two proteins that we want to target. So we want to create a protein that interacts with these targets. And in most cases, what we want is we want to downregulate the target. So we want them to essentially be like sand in the gears of a car, so that actually that pathway we slow down by throwing in proteins that interact with these guys and effectively block, these yellow areas. Remember I showed at the beginning where proteins within a pathway interact. If we throw in a protein that actually sticks to one of the yellow areas, then it changes the yellow areas, and then they don't fit anymore. So the lock and key fit isn't there anymore, and then they can't interact, and that slows down the pathway, and that's what we want. On the other hand, though, we don't want what most of us know as side effects of drugs, right? So the nasty thing about drugs is that often they don't just interact with the proteins in the pathway that we want them to interact with. They also interact with other stuff in our body, with other proteins. And that can create very nasty side effects. So we don't want that, obviously, right? So most medications, they have side effects, nearly all of them. Now, the big new thing with synthetic proteins, because we compute them, is that we can optimize them to only interact with the targets and not interact with the people we don't want an interaction with, which are called the non-targets. That's the goal, right? So design a protein that interacts with one or more targets and doesn't interact with the non-targets, right? And that's effectively drug design, right? So we do an intercept in a pathway. And, aspirin, for example, does just that. It's a small medical compound that happens to fit into one of those yellow areas and happens to slow down the interaction. The problem with traditional drugs like aspirin is that they're fairly small compounds. So the probability of them fitting somewhere else is pretty high. So, aspirin does have side effects. So the idea of proteins is to be really as a drug is to use full-size big proteins, which are very specific, the way nature does it, right? Because our proteins in our body, they have this size because like a key, they have to have enough detail to be specific for a lock. And the term maybe you've heard is called a biologic, this kind of drug, right? And so here's our algorithm. The basic idea of the algorithm, again, I'm not going to bore you with the details. It's a long process. The basic idea of the algorithm is to simulate evolution. So what we do in our drug design algorithm, so we have a target that we want to interact with, and we have non-targets that we don't want to interact with. And typically in most applications, we have one protein as a target and non-target, everybody else, all other proteins, because we want zero side effects, right? So now in our algorithm, we start, we simulate evolution in the following way. We start with like, let's say 1,000 random proteins. And then what we do is for each of those random proteins, we calculate a fitness. And the fitness is a number which represents whether they interact with the target. That's good, then fitness go up. And the protein does not interact with the non-targets. That's bad, fitness goes down. So we do a bunch of math to calculate for each of our candidate a fitness representing whether the protein does what we want it to. So we have 1,000 of these guys. We start with random, and for each calculate the fitness. And now we have a bunch of people who are better than others. And so we do like evolution. The non-fit guys, they die. So we give everybody a probability of survival depending on the fitness. And based on that probability, we let non-fit proteins die. And the ones that are fitter, that are better for us, we let them mate, like in real life. So we take the more fitter proteins and like, let's say we have two proteins that are pretty good. So then we create a new protein, which is half of one and half of the other, right? And that's what's called a crossover. Or we take a relatively good protein and we mutate it and see whether that maybe improves it. So out of that, we create a new generation of candidates. And then for the new candidation of candidates, we do the same thing again. We calculate for everybody the fitness, and then, you know, the non-fit guys die out, and the fit guys mate or mutate. And, you know, some of them that are really good just get copied as is. And now we keep running this method. And it's extremely compute intensive, as you can imagine, because for each individual fitness calculation, we need 20,000 protein interaction prediction using the algorithm I showed earlier, right? So we run this thing on, this really needs now a 40,000 core processor. And we designed a kind of it's not really a drug, but we wanted to try out whether this thing actually works. Again, all of this is nice in theory, but if nature doesn't agree with us, it's useless. So what we did is we looked at a yeast protein, and remember the DNA repair? So we tried to design a drug. It's not really a drug, actually. It's a nasty thing. It's a protein that inhibits DNA repair. It targets a protein called PSK1 in yeast, which is one of the proteins involved in DNA repair. And so by attaching itself to that protein, it will inhibit DNA repair. So what that means is that poor yeast cell that is hit by our medication will now be very sensitive to radiation and will die even under small amounts of radiations. Okay, so this is, so we take, we run our algorithm, and it ran on on 40,000 processors for about 10 hours straight. And what comes out as the best possible protein in our evolutionary algorithm to do what we want is this red sequence here. So it's kind of funny. You have this gigantic machine. After 10 hours, it spits out a red character sequence. It's a little bit like in the movies. And that red character sequence is now our protein, our new drug. And then we sent it off to a lab that actually builds the real protein out of this character sequence. So they actually built the real protein. And then we do our experiment. We dump it on some unsuspecting yeast cells, right? So this is the experiment here. So we do 3 rounds. So number 1 is we just have a regular yeast cell and we put 30 seconds of UV light, of radiation on it, and the yeast cell doesn't really mind. Then what we do as a test is my friend Ashkan actually create what's called a knockout yeast. So this is another type of biochemical experiments where you literally dissect the yeast cell and take out PSK1. So this poor yeast cell is literally physically missing PSK1 protein. And as expected, it will be very sensitive to radiation. And so after 30 seconds of UV light, the poor thing is dead. So now we know that with PSK1, it survives. Without PSK1, it does not survive 30 seconds of UV light. So now our main experiment is we're going to take a regular yeast cell. We're not altering it like in the middle experiment. We take a regular yeast cell, and on that poor yeast cell, we dump our medication, in quote medication, and we dump our anti-PSK1 protein, this red sequence here. And what happens is the poor thing now, its PSK1 protein doesn't work anymore because our anti-PSK1 kind of attaches itself to the PSK1. It's like a stone around its neck. And PSK1 doesn't work anymore. And so the poor thing dies after 30 seconds of UV light. So this is a very simple experiment to figure out these things. And here is the actual experiments, how it looks like. These are the actual yeast colonies. Good. So this was our first experiment to show that our genetic algorithm maybe works, right? It worked for this one. So, well, by coincidence, one of our students was giving a talk like what I'm doing right now at the Ottawa Hospital. And so there's the Ottawa Research Hospital. There's Dr. Blaise. He's part of the muscular dystrophy project. And after the talk, he called us and said, hey, I could meet you guys. So muscular dystrophy is a very serious sickness where the muscles shrink and to the point that people die. And the reason they shrink is because we have these stem cells, these blue little guys here in our muscle fiber, which when muscles get torn, which happens all the time, these stem cells create new muscle cells and, regenerate our muscles. So people who have muscular dystrophy, these stem cells don't work. And so the Ottawa Hospital Research Institute is working on this muscular dystrophy stem cell therapy. And so what they want to do is take healthy stem cells from healthy donors and inject them into people who have muscular dystrophy. So there's lots of issues here. One issue is how to transport the stem cells, because what happens is what's called premature fusion. So when the stem cells get extracted from the healthy person and you put them in your vial, and then the stem cells think injury. And actually the stem cells start creating muscle fiber. That's what you see on the right. So on the left, you see stem cells extracted, but a few seconds later, these stem cells, because they say, oops, there's something bad going on here, we should be muscle fiber and regenerate. But then in your vial, you end up with muscle fiber and you can't really inject it anymore into the sick person. It's called premature fusion. And Dr. Blaise at the Ottawa Research Hospital, he has been studying this fusion process going on in muscle cells. And he identified the proteins that are involved in the pathway in the process of fusion of stem cells. And so there is a protein in particular called 6-1 and another protein called IA. And those two proteins are involved in this fusion process. They're kind of the central guys there. And so the question is obvious. Can we build, similar to the yeast thing I just showed, a protein which disrupts this, at least temporarily? So we want a protein that hooks up to either 6-1 or IA and, and disrupts it. So that's exactly what we did. We ran our method against these two proteins, 6-1 and IA, as a target, right? Everybody else non-target. And so here, and we ran a whole bunch of runs, right? So each of these runs you see on the left is 11 hours on the big machine in Toronto. And so we get a whole bunch of candidates because, the guys in the hospital, they have other criteria also that are important for them, in addition to just attaching themselves to 6-1 or IA. So when they looked at our candidates, they chose these 8 candidates, and they actually ran experiments with them, with these 8 candidates. And out of those, the 2 ones you see here with a blue arrow, the 2 ones that actually in the experiments showed an interaction in the gel, they are listed below. Those are the ones they actually used. And they're working by now on human tissue experiments, and we'll see. So hopefully this all will, the whole stem cell therapy, this is just one problem in their stem cell therapy, but hopefully we have contributed here towards them doing stem cell therapy for muscular dystrophy. The other project, last but not least, I want to talk about is, like everybody else, of course, we're working on COVID-19. And it's a very obvious case, right? So if we all know about COVID-19, we inhale it, and, so up here is the little, picture of COVID-19 virus. And below here, the yellow is the outer membrane of one of our cells, in particular the lung cells. So this is the yellow is the cell membrane of one of our cells in our lungs. And particularly the cells in our lungs, they have a protein sticking out, which is called ACE2. All of our cells have proteins sticking out, which are like sensors, right, for environmental issues. And one of the sensors sticking out, and particularly so in lung cells, is a protein called ACE2. So now COVID has this nasty property that it binds to ACE2. So more precise, actually what you see on the right is, when you look on the TV news, the way they show COVID viruses is not really correct. It's not like a round little ball like they show it. Actually, the COVID virus is here shown on the left. That's really how it looks like. It's kind of ugly, nasty thing. And it has what's called a spike protein sticking out at the bottom here. And at the end of the spike protein, it has a shape of a cup. And this cup is the ACE2 binding site. So this cup actually matches the surface of our ACE2 protein. And that's how it attacks us, right? That's how it attaches to lung cells. And once it's attached, it actually then slides through the cell membrane and gets into the cell. But the main thing for medical purposes is to stop the attachment in the first place before things get worse. So now for us, the obvious question is, can we create a protein that connects to the ACE2 binding site, right? We want to create a little ball, a little protein that is very specific to the ACE2 binding site of COVID. So it goes in here, changes the surface of the cup. The cup doesn't fit ACE2 anymore, right? And that's exactly what we've been doing. So it's similar to the muscular dystrophy project. We ran an awful lot of experiments, computational experiments, and at the end of the day came up with 11 good candidates, out of which we synthesized. We really had the lab build the actual physical protein for 9 candidates. And then using those 9 candidates at the Ottawa Hospital, they tried docking experiments on ACE2. And 2 of those proteins actually interrupted the docking process, right? And those 2 proteins now, I mean, at the Ottawa Hospital, they can't really use live COVID experiments. So they just have the spike protein of COVID, not the whole COVID, because dangerous. And so after the successful tests at the Ottawa Hospital, it went off to Winnipeg. So Winnipeg is the only lab in Canada that is allowed to do actual live virus experiments. They have the right lab to do that. These are the guys who did the Ebola vaccine, right? So they really have a high security lab. So they actually ran a test with our proteins and it was successful. So my colleague Ashkan and I, we are currently actually in the process of publishing this. And we also, we just had 2 conference calls with AstraZeneca. So they're also interested in this. So hopefully, but we want to publish it so everybody gets it, right? The idea at the end of the day is this is not a vaccine, this is a treatment, right? It would be kind of like a puffer. Like when I was a teenager, I had bronchitis, right? And you use kind of, you have problems breathing like you have with COVID, and you use the puffer to open up the lungs. And so this would be similar when people have COVID, they have problems breathing, you puff this into your lungs, and then, you simply get rid of a whole bunch of these COVID viruses and, relieve the lungs. Okay, that's the end of my presentation. Just at the very end, I just also like to, I feel strongly that scientists don't just work in the lab. They also have a duty to society who fund our lab. And I'm really impressed by how the Ottawa police got rid of, the anti-vaxxers and the COVID deniers without anybody being hurt. You see a picture here, like my wife and I, we wanted to go to the counter demonstration, but we were a day late. We came on Sunday at 2, but the counter demonstration was on Saturday at 2. So the 2 of us actually walked, just the 2 of us, past Parliament. And, we saw firsthand that these people are not, people who are genuinely concerned about efficacy of vaccines. They're really just a bunch of rowdies. And they were trying to attack us, and the police had to save us. So to end on a very funny note, this is a friend sent this to me. So the difference between Canada geese and anti-vaccine truckers. Thank you, Frank. Very much.
So Frank, we have a number of questions that I'd like to go through with you. So Your fan, Michael Wolfson, has got a number of questions here.
Whoa.
So his first question was, how much by using machine learning is the analysis ignoring the physical shape, the pattern of folding of the two candidate proteins?
Yeah.
To what extent does the algorithm take account of detailed 3D protein shapes?
Okay, so this is part of our expert discussion here. So the answer to this is that we on purpose try not to work with shape. I mean, there is a lot of different approaches to protein-protein interaction prediction, right? It's an extremely important problem. There's books written about this. And there is a whole group of people who literally use the shape and see whether the shape matches to find out whether two proteins interact or not, right? And so the question comes from that. And that approach is great, but it has a lot of problems in that for many proteins, the 3D shape is not known. So you have to know the 3D shape. So to check out whether it matches. And finding the 3D shape is a very, very hard problem. So we try to avoid that. We try to do prediction from sequence only. And because of that, also now this whole idea of building synthetic proteins would be impossible if you would use the shape for prediction, right? Because we have new unknown synthetic proteins of which, of course, nobody knows the shape. So shape for prediction for our purpose of building new drugs is not possible.
Okay. And then Michael's next question is, could you comment about the specific kind of machine learning algorithm you've developed?
Yeah, I mean, the trick, okay, I can go a little bit more into the algorithm here. So this is the algorithm. So there's lots of machine learning algorithms out there to do this, right? And when you look at, these competition that we were part of, most of these algorithms we're competing with use standard machine learning approaches because you have a database of known interactions and you have a new pair of proteins. And this is really like discovering cats. So you can use support vector machines, decision trees, neural nets. You go through the whole catalog of machine learning methods. And that's what these algorithms do. Problem with it is that it's an oversimplification. Like Ashkan, my colleague, who's actually doing these experiments, he discovered that the binding sites are rather small, 20 amino acids, right? And so the disadvantage, but when you have the data only says protein A and B interact, we don't know where they interact. So the machine learning, the standard machine learning methods effectively get confused by the fact that there is a lot of data that is irrelevant for the interaction. So what we really do here is counting these co-occurrences. We try to hand code our machine learning method to zoom into these little motifs, and we try to learn the motifs that create the interaction rather than looking at the entire protein. And that effectively is the biggest difference. And that's why our method is so much more successful, because again, the interaction between two proteins only happens in like a little window of, let's say, 40 amino acids. And all the other amino acids are effectively irrelevant, but the machine learning kind of looks at them. Okay. I hope that answers it. Sorry, it's a bit long-winded.
Oh, John,
John, I couldn't hear you. Your microphone is off.
You're muted, John.
I guess I should unmute, eh?
Yeah, so now you're back.
No one wants to listen to me.
Yeah.
So Michael says, some years ago, he learned about the so-called interactome.
Yeah.
Is that what you're estimating? And
Yep.
that there's a similarity between that for, say, dogs and humans.
Oh, that's a great
Does this imply
question.
that your algorithm can be trained on data from more than one species?
Oh, absolutely. Like that's what we're doing. Absolutely. So it's just I didn't have all the time in this talk to talk about this, but I'm already kind of showing on my slides a few other organisms, right? Like yeasts and little worms, and you could do dogs and cats and rats and all of them. And so for example, so this is the interactome, right? What he's referring to. It's the graph of the interactions. And absolutely, we have been doing this for many different species. And we're even been looking at using the data available for one species to predict the interaction in another species, like cross species and all of that, right? All different combinations, absolutely. People have written theses about it, and there's lots of papers published on that.
Okay. So another question now from Alexander Smith. Can design proteins be used to increase a deficient pathway's activity? Or is that a much harder problem than reducing overactive pathways?
Oh, very good question. And the answer is we don't know. We haven't done that yet. No. So far, all the problems we looked at was down-regulating pathways. But it may be possible. I don't know.
Okay. And then back to Michael on your in your INSIPS algorithm. Is it an extension of, I don't know, John Holland's genetic algorithms from a few decades ago? I'm unaware of that. Do you know that?
Yeah, it's the standard genetic algorithm. Yeah, anybody who's into genetic algorithm will see that we are following the standard straightforward path here. Yeah, there is no change of the standard genetic algorithm here.
So if your anti-PSK1 synthesized artificial protein has more than one way of folding, is its active site is not always present?
Yeah, that's a very good question. So let me go to the anti-PSK. Sorry, I'm just. Absolutely correct. The question of stability of the proteins we generated is an important one. And natural proteins in our body are amazing in that, I mean, evolution has worked towards, in our body, we only use proteins that are stable in that they always fold into the same shape. Now, when we create a synthetic protein like that, this is not clear. And to be honest, our algorithm doesn't really guarantee that. So for example, when we did the muscular dystrophy, that's why we create that many. So because one of the questions is, when we synthesize them, how stable it is, does it always fold into the same? And we only have a certain hit rate for that. And so that's why we generate a whole pile of them. And then in the lab, we try out which one is stable. So remember here, we chose these ones. Some of those that we excluded were excluded because they were not stable. And they sometimes fold this way, sometimes fold that way. Sometimes they don't fold at all.
And then the last question from Milan Yavor, it says, it seems your anti-PSK1 blocker protein is a powerful toxin.
Yeah.
Is the lab a high-level secure facility to prevent an escape from the lab?
Well, our anti-PSG is toxic for yeast, so it has nothing to do with humans. And well, it would be bad for beer production, I guess, if it got
That's
out.
right. Oh, bread.
into the wild, but it doesn't multiply on its own, right? It's a single protein sitting there. It's not like a virus. A virus is dangerous because it can multiply. A single protein, no, it is not dangerous at all because it cannot multiply, right?
Okay, I think that's, I don't think there's any more questions left. I'll just double check here. Just a couple of people saying thank you for your comments. And I'd like to join the rest of the audience in thanking you, Frank, for this,
Thank you.
this great talk, wonderful talk, and a great demonstration of the need for interdisciplinarity as we move forward. Something that we always talk about, but not necessarily always achieve. So this is really good. So thank you again on behalf of all of us. Thank you very much, and see you soon.
Thank you.
Thank you.
Thanks a lot.
Thank
Bye,
you,
everybody.
everyone. Thank you very much, Frank. That was great. Thanks, John.